So today I'm going to talk about RNA transcription, which is the use of one of the DNA double strand act as a template to produce a messenger RNA, mRNA, which is a temporary copy of a gene that contains information to make a polypeptide. But before going into details, I would like to have a question for you to think about. So we have learned the difference between prokaryotes and eukaryotes. Do they undergo exactly the same transcription process? So think about it. And the answer is no, but the process is very similar. The key points in there are a series of enzymes that perform different functions, and that is very important to remember. Okay, so this picture is the general overview of the three stages of RNA transcription: initiation, elongation, and termination. Okay, so during the initiation stage, for bacteria, the prokaryotes, there is a specific region called promoter, which is recognized by RNA polymerase holoenzyme, and there's a specific sequence called sigma factor that recognizes this sequence, and it forms the open complex. And the RNA polymerase holoenzyme coenzyme synthesize RNA primer. A short strand is made within an open complex, and then the sigma factor is released, then the initiation ends. So for the eukaryotes, it also has a promoter sequence, and the sequence consists of regulatory elements, the tata box and the start site. And there's a series of general transcription factors and a mediator that is involved to open that complex. So it comes to the elongation stage now. The RNA polymerase is just sliding along the DNA strand, and the RNA, the red, represents the mRNA that is newly synthesized. The mRNA is made from the 5' direction to 3' direction. And then we go to the termination stage. Generally, the termination stage is just a terminator enzyme that binds to a termination site, and it causes the RNA polymerase to release and also a completed RNA transcript. Then this RNA transcript is ready to be translated into protein. But more specifically, let's take bacteria, the eukaryotes, for example. It has a role-dependent termination stage and a role-independent termination stage. So the role-dependent stage, it means the role, there's also an enzyme that binds to the RUT, the role utilization site, and the RNA polymerase reaches to the terminator, and the stem loop that causes RNA polymerase to pause, and the role catch up to open complex, and it causes of the components to separate. And for the role independent termination stage, it is caused by a stem loop followed by the U-rich region with help of NUSA protein.